Okay, everyone, so now that we got our heads around diffusion, okay, so remember that diffusion is the movement of any molecule from an area of high concentration to low, okay. Now we can start thinking about osmosis. Now, osmosis is the movement of water molecules from an area of high concentration to low concentration. So osmosis is just a type of diffusion. Right, let's have a look at this concept. This is a really important concept because it's actually going to come up again and again and again with regards to such things as blood, okay? So we need to really get our heads around what this word means. Now, osmolarity is just refers to how much solute you have in a solution. All right? How much solute you have in a solution. So if we have a look at this solution here, we can count the number of water molecules and solute molecules, right? So here in our first beaker, we have 1, 2, 3, 4, 5, 6. 6 out of the 8 is going to be water. And how many solutes? 2 out of the 8 is going to be solutes. Now, what's 6 out of 8 as a percentage? That is a good question. And because I'm terrible at maths, I'm going to have to work it out with a calculator. Wow, that's bad, eh? So 6 divided by 8 equals times 100, I'm so embarrassed, 75%. 75%. And that means I've got 25% solute. Okay? I know, stop, I'm embarrassed. Okay, let's have a look at this solution, shall we? How much water's in there? 2 out of 8. How much solute's in there? 1, 2, 3, 4, 5, 6. 6 out of 8. All right, so 2 out of 8 equates to 25%. Solute equates to 75%. Right, oops. Okay, right. So this particular solution has low osmolarity compared to this one. Because what you're looking at is you're looking at the ratio of solute to water. And this one has a higher ratio, a higher percentage of solute to water. Okay? We're going to do a lot more examples of this, so let's just keep going, yeah? Right. The reason why we need to understand solutes and water and their percentages in a solution is because we can actually make cells shrink and make cells blow up just by using solutions. Okay? Yes, you have heard correctly. I can make a cell blow up just by using a solution. Okay? Just by the solution around it. I know, it's crazy. This ability of changing the shape of cells just by what's around it is called tonicity. Okay? Tonicity. Right, so let's have a look at this example. And it's very, very kind of methodical, the way that I do it, but you guys might come up with a different way of doing it. It's up to you. Okay, So this is my beaker, and here is my red blood cell, okay? How many water molecules are in there? Okay, I can see I've got 2. And how many solutes? I can see I've got 2. All right, so inside the cell, I've got 2 out of 2. Oops, water to solute, sorry. So we've got 2 out of 4 are going to be water molecules. 2 out of 4 are going to be solute molecules. So what do we have here? That means that we have, inside our cells, we have 50% water and 50% solute. Is that correct? Do you agree with that? Yeah. Let's have a look at outside of our cells, okay? Outside of our cells, we have 2 water molecules, 2 out of 4. Solutes, 2 out of 4. That means we have the same thing. Right, so let's have a look and see. Inside the cell, we have 50% water. Outside the cell, we have 50% water. Where is the highest concentration of water? There isn't one. It's the same on both sides, okay? So, if the concentration of water is the same inside versus outside the cell, then what happens is that water will not move. Wait a minute. Will not cause a net movement. Okay? So what ends up happening, because our water concentration is the same on both sides, means that the concentration of water is equal inside and outside of our cells. Now, they are both equal. Now, the next thing we need to think about is what moves, okay? So we've learnt about the plasma membrane, how it's selectively permeable, okay? So that means that water can move through our plasma membrane, but solutes cannot. Okay? Water moves through our plasma membrane and solutes cannot. Okay? So the only thing that can move is water, not solutes. So what ends up happening in this scenario is that you will get a water molecule coming out, but then another one will come back in. And that will happen over time. But there's going to be no net movement of water molecules. Okay? No net movement. When the solution looks like this, it's called an isotonic solution. Okay? And the reason why we need to learn this is because intravenous solutions are isotonic solutions. So you're going to be giving these solutions to people. So you need to understand how they work. Okay? Let's do another example. Okay, so let's look at another example, another type of solution. So let's have a look at our beaker here. Let's have a look at the number of water molecules inside our cells. Okay? So we can see that inside the red blood cell, there are 4 molecules, okay? So 2 out of the 4 molecules are water, because they're the blue ones, and then 2 out of the 4 molecules in our cells are solutes, okay? So that means that in our cell, we have 50% water and, whoops, and 50% solute. Let me just make this one there. Okay, let's have a look outside of our cell, okay? Let's have a look. Water. Well, let's have a look at how many molecules we have to start off with. We've got 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13 molecules, okay? So out of those 13 molecules, 4 of them are water and the rest are going to be solute, correct? So 1, 2, 3, 4 are water, and then 1, 2, 3, 4, 5, 6, 7, 8, 9 out of the 13 are going to be solutes. Let's calculate this as a percentage. 4 divided by 13 times 100 is 31% water. And I've got my calculator here. 9 divided by 13 times 100 is going to be 69% solute. Okay? Where is the highest concentration of water located? Here. This one, 50%, is higher than 31. Okay? So there's a higher concentration of water inside of our cells. Okay? So from inside. Now let's think about what moves. Water moves through our plasma membrane, no worries at all. Solutes can't. So that means that water is going to move from inside our cells to outside, because osmosis is the movement of water from an area of high concentration to low. So what happens is that we get our cells shrinking. Okay? So if we put a red blood cell in a hypertonic solution, this red blood cell will shrink. Yeah. I know it's a lot to kind of. So there's a lot of things that you need to think about. Water molecules, solute molecules as a percentage. Where is the highest percentage of water? And then water is the only thing that moves. The solutes don't move. And water is going to move from an area of high concentration to low concentration, because that is osmosis. Okay? So these are hypertonic solutions, okay? And you use them clinically to get cells to draw water out of cells, okay? Right, let's do the last example. All right, let's look at a different solution. So you're going to learn about 3 solutions: isotonic, hypertonic, and hypotonic. Okay, hyper means high, which means too many solutes. Hypo means low, which means low solutes. Iso means the same, which is equal solutes. Okay, let's do this example. Here we go. Inside our beautiful red blood cell, we have 2 out of 4 and 2 out of 4. So that means, again, we have 50% water and 50% solids. Okay, let's have a look at outside. How many molecules do we have to begin with? We've got 1, 2, 3, 4, 5, 6, 7. 7 molecules, and 5 of those are water. That means that 2 of those are going to be solid. Let's have a look as a percentage. I've got my calculator here. 5 divided by 7 times 100. 71. So 71% is going to be water. And that means that 29% is going to be solutes. Okay? Let's have a look and see. The next question we have to ask ourselves, where is the highest concentration of water located? Here. Because inside our cells we have 50% water. In here we have 71% water. So, this is where the highest concentration of water is. So, what moves? Water or solute? Water moves by osmosis. So water's going to move from an area of high concentration to low concentration. So it's going to go from here into here. Yeah? So that means that water is going to go into our cells. And that is what happens. And if we allow enough water to go into our cells, the cell can only cope with so much and eventually go pop. Okay? So this is the solution that you will use. Okay? All right. You're going to have lots of practice dealing with isotonic, hypertonic and hypertonic in order for you to understand that crucial concept in your tutorial activities. Okay? So this is the result. Isotonic solution, no net water movement. Water goes in and out, but no net movement. Hypotonic solution results in water gain. Hyper results in water loss. Okay, so let's move on now to something else. So now let's just get our heads around what active and passive transport means, just to finish off this little section. Okay, so we know that things go through the plasma membrane. Okay, but there are different ways that things can move through the plasma membrane. Okay, now there's 2 major categories. All right, one is called active and the other one is called passive. All right, now what you guys need to understand now, and we will see a lot of examples of this. One requires energy and one doesn't. Okay, the active transport requires energy, which comes from which organelle? Did you say the mighty mitochondria? I hope you did. And one is passive, that does not involve energy. That's all you need to know at this point. And there's also other ways that we can move things through the plasma membrane. So the plasma membrane, you can go through it via diffusion. You can use a protein, okay? So if you use a protein, you can use energy. If you use a protein and you use energy, it's called active transport. well, if you use energy, doesn't matter if you use protein, it's active transport, and then passive transport is, okay, so if you don't use energy, it can be with or without a protein, okay? So there are different ways that we can get through this plasma membrane. One is just diffusion, other way is using a protein, and another way is using these bubbles or vesicles, okay? This is how we move lots of stuff at once, okay? We just package it up and off it goes into the plasma membrane, or the plasma membrane makes a little bubble and we just grab the stuff from the outside and then it pinches off and it goes into our cells. When you look at these vesicles, it's just like when you blow a bubble under the water, okay? The bubble goes all the way to the surface and then it pops and it releases the air on the surface. That's exactly the same way as how these endo and exocytosis is. Exocytosis is like having a bubble of air under the water. It goes up, plop, and off it goes. And endocytosis would be the reverse. It's like you're grabbing air from the outside and bringing it into the bubble, into the water. Okay? I'm being very animated today. Very, very animated. All right, I hope you enjoyed that. We'll see you in the next recording, hey?